Swayam Prabha. Digital India. Educated India.
We welcome you all to the next class on Inorganic Chemistry of Life Principles and Perspectives. In the last two classes we have started looking at the zinc containing enzymes. In the first class I have talked to you general classes and then followed by the example of the alcohol dehydrogenase explained to you fully. Just in the previous class I have taken two other enzymes. Both these enzymes are based on the adding water to carbon dioxide, converting the carbon dioxide into the H2CO3 minus in order to balance the acid base components of the cell, of the tissue in the body, organs, etc. And the second example I talked to you about the peptide hydrolysis, particularly the peptide hydrolysis. I have talked to you in more detail the carboxypeptidase, which means that this particular carboxypeptidase enzyme can only act on the carboxyl terminal of the polypeptide, but not the N terminal. And secondly, I also try to impress you on this enzyme recognizes the last two residues, and particularly depending upon the side chain of the last residue, the enzyme will work. If it is carboxypeptidase A, it will work on hydrophobic kind of a side chains, and if it is a carboxypeptidase B, it will work on the cationic kind of species which are there in the side chain, and if it is a carboxypeptidase C, it will be acting on peptide having a side chain of the negatively charged. So, therefore, it is very selective. So, therefore, also you have a huge number of peptidases are there. Similarly, I also mentioned we have amine terminal cleavage, etc., and other types of things. We are not going into the details. Now, the next story is just to see we have looked at the oxidoreductase, we have looked at the hydration or the condensed bond breakage. Now, let us look at some kind of a transfer. So, you take some group from one and put into the other. So, example here shown here is a betaine homocysteine methyl transferase. It basically transfers the methyl moiety. As you can see over here, this is called the glycine betaine. So, it will go into N,N-dimethyl glycine. So, this will go into N,N-dimethyl glycine, and this is coupled with the homocysteine going to methionine. So, what is the difference between the homocysteine and methionine? One of the methyl group is going onto the sulfur. Cysteine SH, methionine SME, that is you keep in mind. So, cysteine is SH and the methionine is SME. So, therefore, you are basically transferring the group, one of the methyl group over there. So, these two are coupled together. So, that is what is mentioned here. It is involved in the regulation of the homocysteine metabolism. It converts the betaine and homocysteine to dimethylglycine and methionine respectively. So, you can see that the glycine betaine will go to N,N-dimethylglycine, and on the other hand homocysteine will go to the methionine, okay? And that is what we are talking about. So, this is basically a homocysteine metabolism. In the process, the metabolism takes place when the methyl group is transferred. In this enzyme, there is a catalytic zinc in this BHMT is a short form for betaine homocysteine methyl transferase. Betaine homocysteine methyl transferase is referred as BHMT. So, in this case, this is the enzyme. In this the structure of the enzyme you can see over there. Okay. And in this enzyme, the catalytic zinc of this one is bound by 3 thiolates and a hydroxyl group. And there is a rapid rearrangement of this enzyme during the reaction when it goes. The enzyme undergoes oxidative step and enzyme undergoes reductive step, but zinc 2 plus does not undergo redox. So, this is again another case. So, there is an enzyme undergoing reduction oxidation, but zinc is not undergoing any reduction oxidation. So, here what is going on? It is the cysteine and SH, and disulfide is SS. So, what is the cysteine and disulfide relation? Cysteine to disulfide relation is, cysteine is let us say cysteine SH, and so two of such things can give cysteine SS. So, this is oxidized form and this is reduced form. So, in this manner the enzyme can undergo redox. So, this is the betaine homocysteine methyl transferase. Betaine homocysteine methyl transferase. So, it is basically that one. In this particular reaction there is a redox occurring. The redox is not occurring by the zinc, but occurring by the cysteine to disulfide. So, a cysteine bond, disulfide bond is formed between the two of the 3 zinc sulfur binding ligands. As we have seen mentioned that there are 3 cysteines are there, and these 3, out of the 3 cysteines and the zinc, 2 of them will form a disulfide in the oxidized form, and in the reduced form they convert to the coordination. So, zinc having 1, 2, 3 cysteine and hydroxide S cysteine in the reduced enzyme, and in the oxidized enzyme this will, zinc will move away, and this one of the S cysteine and other S cysteine, this will form a disulfide bond, and this moves out from here, okay, and this is S cysteine and OH. So, this is the reduced enzyme, the other one is the oxidized enzyme. This is not the reduced one, this is the oxidized system. So, this is an oxidized enzyme, reason is SS bond is there, and this is the reduced one, the SS is broken to S cysteine and cysteine thing. So, this is so you have on the left side is the reduced enzyme, on the right side you have the oxidized enzyme. So, but in both the cases zinc is zinc 2 plus, there is no difference. Okay. So, I hope this is clear in this particular system what we have here, betaine homocysteine methyl transferase. So, let us look at a bit more details. So, whatever I mentioned to you, that you have a reduced enzyme where the zinc is attached to one cysteine, another cysteine, another cysteine, 3 of them. This is oxidized enzyme, only the zinc part is not shown here, zinc will go somewhere, and then you have seen that the 2 of the cysteines have become disulfide. So, the cysteine 217 and 218, this will form a disulfide bond. Okay. So, reduced form, oxidized form, this is how it goes in these ones. And as a result, how does this happen? This when this happens, the cysteine 217 should rearrange, okay, to form a disulfide bond, okay, and sulfur moves in this rotation. So, you have a great change in this particular conformational region of this. Okay. So, this is the replete enzyme where the zinc is present, oxidase enzyme, zinc is out of the coordination sphere, and that is what you call oxidase enzyme and replete enzyme. So, in this cysteine 217 is the one which is, that is somewhere here, this is the one 217 rearranges to form a disulfide with the 299. So, the 299 is with this one. So, it will form a bond with the disulfide bond. So, the sulfur of the 217 moves because it has to come closer, the 2 sulfur atoms have to come closer, and for that there is a rotation about this bond, and this kind of sulfur of the moves by rotation about the C alpha C beta of the cystenyl moiety, and then say flips the 217 to 80 peptide bond flips in order to accommodate the disulfide bond. So from this kind of a coordination sphere to form a disulfide, because two of the sulfurs have to come closer, this has to go away, therefore, there is a movement of this bond and these two coming closer and then forming a disulfide and throwing out this away. Okay? And that is where the thing is there. And let's look at another issue which I have not brought to the notice to you earlier, but now I will bring. This is the last structure. What is this one? So this is nothing but the homocysteine glycine butane transition state. So you have to, you are forming the transfer of the methyl group. Before that, there is a kind of a intermediate step. You can see that intermediate step. Once the intermediate step is they going to the reaction, then the methyl transfer will take place. Okay. So, and when in that kind of a case, you will get in one case N,N-dimethyl glycine, and in the other case you will get the methionine. So, if the methyl group goes over here, this will get the methionine and this will become N,N-dimethyl. So, this is an intermediate. So, first the two the basic components will bind to the protein or the enzyme accordingly by the recognition process, and then form an intermediate or form a transition state, and you can see that is the kind of thing. This is still long bond, and once this bond is shortened, the methyl will be transferred over there, and this whole thing is bound in the region of the zinc containing one. Okay. So, as you can see, this is one of the view, this is a perpendicular view to that, and that is how your molecule of the intermediate, the two substrates joining together maintaining this one is bound. So, this has to have be very close to the zinc as well as the disulfide formation. So, redox is so here you can see homocysteine, okay, and the hydrogen bond interaction are important for stereospecificity. You can see that this is the part of the homocysteine which is interacting with this. This is, we call it as a recognition. So, in any of the enzyme reaction, the substrate recognition is absolutely essential. So, that is what you are trying to see, that part of it is. And this is another view of this, a view which is about perpendicular to the previous one, which is 90 degrees from that, showing the. So, this will show the carboxylate group of homocysteine analog of the carboxylate group, that is that is the glycine butane that is interacting. So, you could see both the things. So, you see here, this is the carbox, this is one end, you are looking at these ones, in the other end 90 degree you are looking at this one. So, that means this particular the two substrates which are bound over here are basically recognized by a variety of hydrogen bonds around the zinc center in the protein. That means when this comes in, a lot of conformational change is important, and that happens, and that leads to this particular transition state. This is basically a transition state species, and then when it becomes shorter, then it will become the regular species. So, the same thing which is shown over there for the carboxybutyl homocysteine binding in this, both the things, and then it will undergo. This is not very important because I have already explained to you. Okay, the zinc site and changes accompanying the oxidation and the loss of zinc, and this is the A, and that means zinc is basically out of the coordination sphere. That is what it basically means is. This is basically a simulated annealing omit map that will show you where the zinc supposed to have been there during the displacement, and this is electron density map in the vicinity of the zinc in the reduced enzyme. Now, in the B, what you have is the geometry of the metal cluster, as you can see, metal ligand cluster, and the interaction of the cysteine sulfur with the residues is 216 and 218, and the water are indicated by the dashed kind of a species over here in lines. So, the water is also hydrogen bonded to the backbone of NH of the phenylalanine 31. So, the 4th sulfur is from the bound analog of the homocysteine, this complex of this. So, that is, so first of all it should bind to the 2 parts of the thing, and this is recognized, and then the transfer takes place. In the transfer is also triggered by the redox, and the enzyme goes from the reduced to the oxidized, and that is happening not because of the zinc 2 plus undergoing anything, but it is because of the cysteine is undergoing, 2 of the cysteines are undergoing to oxidation of the disulfide. And this is how the transferase reaction, betaine homocysteine methyl transferase. And as you can see at the end, in this process the glycine betaine will go to NN dimethyl glycine, and the homocysteine will go to the methionine, and this is what is exactly happening in this. Now, let us look at another kind of property or function of these enzymes, ligase. Ligase means basically a kind of a repair. From the 2 pieces make a bond. So, binding 2 pieces together is the ligase activity. So, this ligase is involved in basically DNA repair mechanism. When the DNA is cleaved either in the single strand or it is involved in the double strand, there are different kinds of ligases are there. So, these ligases will repair this particular broken piece. So, basically by catalyzing the formation of the phosphodiester bond. So, this will help in making the phosphodiester bond in this particular thing. So, therefore, it is basically a repair kind of a thing. It can be doing the single strand breaks, it can do, or it can do the double strand break, that there will be different kind of thing. So, repaired by the DNA ligases using the complementary strand of the double helix as a template. The DNA ligase creating the final phosphodiester bond in order to fully repair the enzyme of the DNA. So, as you know very well, in the body there is continuously the DNA cleavage keeps happening, the ligases act on this and repair. So, that is covered. So, this happen because of the chemical assault, it can happen because of the radiation assault, it could be sometimes UV radiations can also create this one. So, DNA ligase is used in both repair as well as replication also it is there. Okay. One other system which is used in the DNA binding I will show you in the later on, but right now let us look at this called zinc fingers of the things that I will show you in a while. So, the purified DNA ligase is used in gene cloning to join the DNA molecules together to form a recombinant DNA also. So, it is absolutely involved in that. So, as you can see, the principle only I will explain, I will not be going into the more details. So, do not bother about more details of this. Now, you have on the left side 3' system, here on the right side the 5' system, and these two joining together. So, when the hydroxyl and the PO thing join together, what will happen? Water will be lost because it is condensation, and a diester bond of the phosphate is formed. So, in this case it will be 3', 5', because one of the end is coming from 3', the other phosphate end is coming, hydroxyl end is coming from the 3', phosphate end is coming from the 5'. Therefore, when these two join together, you will get a 3', 5' phosphodiester bond. And this is being, when this reaction happens, it will consume the ATP, and the ATP will lose out phosphate as well as the AMP, and that is what you are seeing, one on this direction, one on this direction. So, this is a energy process, it requires a lot of energy in this. Okay. So, therefore, you can see all of them, the 3', the 5', and the ATP utilization, and then finally bond formation. So, you have a 3' strand and the 5' strand together. So, this will lead in the formation of the 2 covalent phosphodiester bonds between the 3' hydroxyl ends of one of the nucleotide, which you called as a acceptor, and the 5' end of the thing, which is phosphate, which is called donor in this case. In this terminology, this is called the acceptor, this is called the donor, and then you will find. So, therefore, adenylation, that is addition of AMP of a lysine residue in the active center of enzyme will happen because the AMP is coming out. This will make that one. So, pyrophosphate is released out of that, PI, PPI, etc., pyrophosphate. So, the transfer of AMP to the 5' phosphate of the donor through the formation of a pyrophosphate bond. So, formation of a phosphodiester bond between the 5' of the phosphate side and 3' of the hydroxyl side, this will form. So, this is the kind of a way, it is a principle based, only the principle based. So, you are making the bond. So, these are how you actually repair. So, initially convert into the adenylation, etc. A is adenosine, NM is the ribosyl nicotinamide, and these are have to bind to the enzyme and then do the reaction. So, I would say you do not need to go more into detail of this, but you better understand this principle part of it. It is only just the principle part of it. So, this can happen in the way that is explained under A, this can happen under B, this can happen under C. So, there are very many ways of pairing the DNA, and please do not bother about all these details of it. The main thing is that the two ends of the broken one are recognized by the enzyme. So, one is the hydroxyl, other is this one phosphate, and then these two are brought together and these two are stitched together, and that is what the principle, if you understand, is good. So, that means zinc enzyme is involved in a ligase property, in replication property, in DNA binding property, all of these kind of things. One another system is also bound to the DNA, DNA protein interactions is the zinc finger motifs. As you can see, there are beta sheet, there is alpha helix, it's a kind of a joining between the beta sheet and the alpha helix. So, this is observed in a large number of transcription factors. So, these can bind very nicely with the DNA. So, helix and beta sheet topology. So, where you have a helix and beta sheet are orienting in this way, and the histidines and cysteines are coming in the proximity, there you will get a zinc finger. So, one alpha helix connected to the two antiparallel beta sheets. So, cysteine 2 times, histidine 2 times are present to the zinc binding center, zinc fingers, found in 2% of the human genes. So, a lot of human gene binding things. So, this is basically used in recognizing the DNA by protein DNA interactions. So, what you have a tetrahedral binding site, you have a zinc at the center, 2 histidines and 2 cysteines are binding. So, it is a zinc cysteine twice, histidine twice. So, you have slightly different variation, cysteine X, histidine 4 minus 1, these are all known variety of depending upon different kinds of enzymes. So, this is one of the important factor. I have not explained you any function on this, just explain these kind of things. One last enzyme I would like to cover for you is that it is not only hydration, not only oxidoreductase, not only the ligase property, we can also have isomerase, isomerizing some groups. And the example taken is mannose phosphate isomerase. That means it isomerizes the mannose moiety. So, this converts mannose 6 phosphate into the fructose 6 phosphate. It converts mannose 6 phosphate into fructose 6 phosphate. Okay. So, basically you have your zinc center in the enzyme where the substrate binds, and that is mannose 6 phosphate, and the substrate interacts through its C1, C2 carbons or C5, C6 carbons here through this zinc center, and that triggers the conformational change as well as the reaction. The neighbor imidazole or histidine will trigger the reaction by the protonation, deprotonation steps of this, and this will form an intermediate, and then this enzyme will work on this mannose 6 phosphate or fructose 6 phosphate depending upon which direction you are going, and finally you get back to this. So, this is what this is nothing but an isomerase. So, you are isomerizing the centers, you can see that. So, you are converting the mannose to the fructose kind of a situation. So, enzymatic reactions are taking place in these ones, you can see that. So, you can see that these two OH, H in this one. So, this will be opened up. When it is opened up, you will see the difference between the mannose part of and the fructose part of that. So, say mannose 6 phosphate. So, for this it has to bind, it has to have a deprotonation kind of a step from over here, and therefore it will rearrange, because this will take into a double bond system, and that will basically take the negative charge over there, and that is what brings in the rearrangement to this. So, so you have enzymes making into this kind of thing. Okay, so what all we have seen in the zinc enzymes, we have seen there are 200 different kinds of zinc enzymes are present in the human body. Some of the reactions are water addings, and this can also do the condensed bond breakage or peptide breakages. You can also do molecules to undergo redox, like between alcohol to aldehyde, aldehyde to alcohol, oxidoreductase. Then we have also looked at the lyase is the water, adding water to the carbon dioxide. Then we looked at the transferase where the methyl group is being transferred, during which again the enzyme undergoes redox. There is this redox is not at the zinc not by the zinc ion, but by the zinc bound cysteines. There are 3 cysteines which are bound in the reduced form of the enzyme, and when the enzyme oxidizes, 2 of these cysteines will come out and form a disulfide bond and pushes the zinc ion out. So, in the process that you have a redox of the enzyme, and then the substrates are bound, betaine, glycine and homocysteine. So, therefore, between these two there is an intermediate is formed, and the methyl group is being transferred. It is very clear. Then one other enzyme that I have explained to you is the ligases. In the ligase is that you have the 2 other bonds, the hydroxyl and the phosphate end, 3' and 5' ends are joined together. This is highly useful in repairing the DNA, where there is a break has taken place. So, therefore, enzyme has to recognize both the terminal of the repair of the broken compartments, and then thereby bring them together and make the join together to form a bond. It can form single strand case scissions. It can also rectify the double stranded scissions or breakage, all these together. And in this ligases, I have not given any kind of a mechanism, I only just give how to visualize, that is good enough. And isomerase, so there is one more enzyme here which I have not shown here, is the isomerase. So, the isomerase basically isomerizes the carbohydrate position that meets with the mannose towards that of the fructose too. For this, what is required? The mannose has to bind at the 5,6, and there is an attack by the neighbor, the histidine imidazole part of that, which is involved in the deprotonation of that, and this converts the carbon center and then brings a rotation for this, and this particular change brings the change in the carbohydrate moiety. So, thus we have seen hydrolysis, lyase, peptidase activity, then oxidoreductase activity, we have seen the ligase activity, we have seen the transferase activity, we also have seen the isomerase activity. So, that brings to a conclusion of zinc based story, and in the next class probably and hopefully I will take up the enzymes corresponding to molybdenum, and then we will try to continue on that for some couple of classes or so. Thank you very much.